recorded and live on YouTube. The recording will be saved on our YouTube channel right after the session. An evaluation survey will appear at the end of our session. Please take some time to provide your feedback as it would be really meaningful for us to improve our next session. Thank you very much for your attention, and once again, welcome to our webinar organized by AIMS Biotechnology Students Association. My name is Renu Priya, and I'm proud to tell you that I'm a biotechnology degree student at AIMS University. It is a great honor for me to serve you as the MC for this webinar. If anyone has any issues with the audio or video, please use the chat box and we will do our best to help you. To start the session, I would like to invite Mr. Hugo, the ABSA president, to deliver his welcoming remarks.
Thank you, MC, Ms. Renu. Hello, everyone, and greetings from Malaysia. Our honorable speaker of the day, Professor Dr. Kalaimati from Florida International University of United States of America, the Dean of Faculty of Applied Sciences, who is also the moderator of this webinar, Professor Manikam Ravichandran from INS University of Malaysia. Professors, lecturers, ladies and gentlemen, thank you for joining us. On behalf of the Faculty of Applied Sciences of INS University and INS Biotechnology Students Association, or ABSA, I welcome you all. I'm very delighted to see all of you here, though it's virtually. This is our 11th webinar as a part of our ABSA webinar series. As an association of students, ABSA always strives to provide a quality and comprehensive social platform to our biotech and bioinformatics students here at IMS University to help them to learn new things, to grow, and to have fun in the process. In this age of modern technology and the ever-growing field of biotechnology, our intellectual capacity is essential. However, our soft skills are equally important. In fact, I would say that our soft skills are more important than ever before, no matter whether you are a researcher, entrepreneur, patent agent, or writer after graduation. Therefore, ABSA has started this webinar series. All members of ABSA will have an opportunity to organize a webinar as a part of the ABSA webinar series. So our series aims to provide a platform for the students to develop and refine their various soft skills, such as leadership, teamwork, communication, interpersonal skills, event organization, and time management skills. These webinars are really meant to be student-centric and helpful for involved students in developing and sharpening their soft skills. While at the same time, these webinars are helpful not just to the ABSA members, but all participants to update themselves with the latest knowledge in the respective areas of webinar topics. Here, I want to thank our prestigious speaker, Dr. Kalaimati. In fact, we are honored to host you, madam. Thank you for accepting our invitation and becoming a part of our webinar series. I would also like to thank our Dean of the Faculty of Applied Sciences, Professor Manikam Ravichandran, for the continuous support on ABSA activities and serving as the moderator for the webinar session today. Then I would like to thank the organizing committees for doing a marvelous job. The efforts, the time they committed to this webinar are appreciable, especially during trying time like this, where face-to-face meeting is essentially lacking. Well, last but not least, my special thanks to our ABSA advisor, Mr. Jivendran. His support meant a lot to us. Ladies and gentlemen, once again, thank you for joining us. Without further ado, Ms. Renu, over to you.
Thank you very much, Mr. Hugo, for the wonderful welcoming remarks. Next, I would like to introduce the moderator of today's webinar, Senior Professor M. Ravichandran. It will be a great pleasure to give a short introduction about our moderator for today, Senior Professor M. Ravichandran. Currently, Professor M. Ravichandran is serving as the Dean of Faculty of Applied Sciences of AIMS University, Malaysia. He completed his Master's in Medical Microbiology from Christian Medical College, Vellore, and gained his PhD in Biotechnology degree from Anna University, Chennai, India. His research interests are genetically engineered cholera vaccine, vaccine formulation, molecular diagnostics, and phage therapy. He has 70 publications in international journals and has supervised 40 postgraduate students, filed 8 patents, and commercialized 3 products on diagnostics. He has been awarded 44 national and international awards for academic and research excellence. He was an associate member and the committee member on Top Research Scientist Malaysia of the Academy of Sciences Malaysia and was also a former Vice Chancellor of AIMS University Malaysia. Ladies and gentlemen, please get ready to have a scientific feast for the ears. And I would like to request the moderator, Senior Professor M. Ravichandran, to take charge of the webinar and to introduce the speaker for today, Professor Kalaimati.
Okay, good day to everyone. Good morning to good evening to Prof. Kalaimati, and good morning to everyone in Malaysia. And we also have people watching from India. And I think ABSA channel is very popular. I think so many different country people will be watching. This talk is simultaneously delivered via Zoom and also via YouTube. So first of all, my thank you to Prof. Kalai for kindly agreeing to give the talk. As an adjunct professor of AIMS University, I think this is the first event we are organizing. Sorry if some mishap happens. I think it's all run by children or students, and hope you understand. I think we all are learning process. And this whole webinar will be very formal because each student have a role to play. I think we are the part of mentoring and organizing it and learning the part of it. I would like to thank ABSA, Mr. Hugo, for kindly taking up this challenge, though it's a Raya holiday and they are everyone in the holiday mood. And though we are in MCO, movement control order, and every student, children are very active and participating in this. So I think I've known Prof. Kalai for a long time. We have been interacting. I saw her bubbling activities one of the Pseudomonas conference in 2019. So I had a chance to meet her. I was really inspired by her way. She inspires student and mentors student, how she move with people, how she mobilize things done. And she is a big game changer in the field of microbiology, and particularly motivating youngsters how to grow, how to, she does a lot of charitable work, and I can talk and learn the whole seminar. I don't want to waste my time. I think Prof. Kalai, we also had a meeting, very fruitful meeting with the Vice Chancellor, current Vice Chancellor of AIMS University. It was very, very fruitful. So I just briefly tell about Prof. Kalai. I think we'll share with you. And I think Prof. Kalai is she done her BSc and MSc from University of Malaya. She hails from Ipoh and learned from Tamil school. And she went to US and did her PhD and MPH, and she did her postdoctoral in Tufts University in Boston, and subsequently in UT Memphis, Copenhagen, Denmark. Endowed with a positive outlook and a keen intellect, armed with sheer determination, she beats all odds to be where she is today. And her footprint is there globally. I think she has connection everywhere in the world. So in 1999, Prof. Mati joined Florida International University, which is the 4th largest university in the US. And they have 277 program, and they have ranked very well in research and so on. It's a very reputed university, Prof. Kalai hails from. And she also spent some time in Harvard Medical School and as a visiting professor. And she, in 2010, she became the professor of molecular biology. And she has mentored more than 150 individuals from school children and professors and so on. I met a student in 2019 in a conference, and she's a Fulbright scholar working next door with her. She hails from Coimbatore, India. And she's been awarded many awards. She also one of the awarded President Council World Ahead Faculty Award, the highest honor at FIU. I think you have to is one of the fourth largest university. She received the highest honor from Florida International University as a President Council World Head Faculty Award. I think we are indeed very privileged to have Prof. Kalai here. And she's also inaugural recipient of NEB, New England Biolabs. Actually, that is one of the pioneer molecular biology company in US where the restriction enzyme bond came from. So she also received NEB's Passion in Science Award for her exemplary scientific mentorship and advocacy. And her heart goes to Pseudomonas. She does a lot of research on Pseudomonas. Anything you talk about Pseudomonas, therapy, resistance, genomics, and most importantly, she's multi-talented. She's not only microbiology and molecular biology, she's also a bioinformatician. She has learned and she does a lot of bioinformatics work and so on. And Prof. Mati has her focus on global health, and she did a sabbatical in WHO, and she has published more than 150 quality publications and 120 invited presentations. And her impact factor, she has published in a highly reputed journal like PNAS and Review of Microbiology, NAR, Nucleic Acid Research, and so on. Her H index is 29. And most importantly, for her seminal contribution to science and the community, Mati has been the first FIU faculty elected for the prestigious Fellow of American Academy of Microbiology. Of the two Malaysian inducted, she is the second and the first woman who was also educated in Tamil language. So she's proud to speak, and so this is one of her biggest achievement. And the Academy is the honorific leadership group within the ASM, Academic Science, American Society for Microbiology, one of the world's oldest and longest science societies. I think there's so many things are there. I think let's enjoy her talks. It's very interactive. Don't feel shy. It's a time to talk, and I think we might take a little bit extra time. I think it is night time at, it's actually 9:15 at US in a place. And thank you for your time, Prof. And I think I'll leave it with you. I'll leave it to the MC, Mr. Hugo, and all the team.
All right. Good evening. Thank you, Prof. Ravi, for the long and lengthy introduction. I hope to do justice to that introduction. Thank you, Renu Priya, and thank you, the president of this student body. I'm glad that you students are very active. That's very important for your, like you said earlier, soft skills are as equally or even more important than all your grades. With that said, I have a long presentation, and I believe I have 45 minutes or 1 hour. I wasn't told how long is my talk.
Time, Prof. You can take it. I want longer, better.
Okay, so well, I try to give a talk around 45 to 50 minutes and set aside a lot of time for questions, and I can share more information after my major presentation. If you have any questions, I do have a few people lined up here to answer on the chat. Because I don't like to look at chat while I'm giving a talk, but I would definitely, after the presentation, we'll look at all the answers, we'll review the answers, and I'll give you a revised version to then will be a link to the YouTube talk. So that way, I want to make sure that the information shared online on the chat is validated. So with that said, thanks. Good to see a lot of familiar faces in the group here, and hope this will be a good session, an educational session for everybody. Would you please allow me to share my screen? I guess I can.
Yeah.
So now the $1 million question is, when I play my slide, can you see that it
Yeah,
is
it's simple.
okay? You see the slide? Can you tell me if the
Beautiful.
slide is changing?
Yeah, it's changing.
Perfect. Okay. So today's talk is on SARS-CoV-2 vaccines. So I will uh, so I give me a few minutes to just to talk about the society that I represent. It's the Microbiology Society, which is based in UK. This is previously known as Society for General Microbiology. And for those microbiologists, this was the society founded by Alexander Fleming, and he served as the first president. Next to American Society for Microbiology, um, this society is the second largest, and student membership is very little, it's only 18 pounds, but then postgraduates is rather high, 41 pounds. Faculty is 82 pounds, but if you show that your salary is less than 35,000, and so the price drops. So if you're a member of the society, you can apply for grants and more. And also it's also free to publish. The society itself has several journals, 6 journals. The last journal is open access, which is it costs money to publish in that open access journal. The rest are free. And the journal that I represent, the Journal of Medical Microbiology, has 8 to 9 subsections. As you notice here, antimicrobial resistance, clinical microbiology, disease diagnosis and diagnostics, microbiome and microbiology, ecology and health, molecular and microbial epidemiology, One Health, prevention therapy and therapeutics, medical mycology, a new group. Now we have a profile, so what we call mini reviews. The editors, section editors, come from all over the world. So do consider submitting articles to Journal of Medical Microbiology. Again, I would reiterate that it's free to publish in this journal. So another disclaimer. கற்றது கைமண் அளவு கல்லாதது உலகளவு. For those who don't know what it means that what we know is very little, what we don't know is as big as the ocean. So the only difference is that maybe I know little more than you do, and some of you here may know more than me, but together we're going to help each other and teach each other and learn something. So here's my outline of the presentation. I want to talk about vaccination from the historical perspective, because vaccination is not new. So I'll start with smallpox and variolation, and then we'll talk a bit into the vaccination and immune response, the types of vaccines, and where are we with the preclinical and clinical research. Then get into SARS-CoV-2 vaccines, and I will specifically focus on the vaccines that are approved not approved, has been approved for emergency use in Malaysia, and a little bit more about that, and then get into the vaccine politics and COVID burden. And then I will end the presentation with why people are so hesitant about vaccination or vaccinating, and how we should be able to prevent that, or try to promote that people do use the vaccine. Okay, so with that said, let me get into the meat of my presentation. And so if you can't talk about vaccine without talking about smallpox. Of course, probably none of us, even including me, have seen a real case of smallpox. It's a devastating and disfiguring disease with 30 to 40% fatality, and the infection is simply by contact and inhalation, and it's caused by a DNA virus called variola. There is no cure for this virus, only prevention. And so this disease is an age-old disease. The early writings on people getting vaccinated or variolated comes from a Chinese history, where this gentleman just blowing scabs from smallpox into nose of a uninfected patient. And if you look at the history, that's the process called the variolations. And then in the 1500s, there's a history of how people take the scabs from the from smallpox and introduce it into healthy people's skin. And then, and you fast forward, even in 1700s, the U.S. doctors noted on the slaves they were that these individuals had this little bump, and then they were told that they had variolation. And the most famous story comes from 1718, Lady Montagu, who is the wife of the British ambassador in Turkey, who learnt about variolation and found herself with a son who was very ill, and she famously variolated him and took the story to UK and did an open variolation. And the important thing to note is that you could, even with a vaccine, you could have a fatality. Right, people still died. About 2 to 3% of the people who got the variolation died. But imagine if they hadn't taken that vaccine, then there would have been a fatality rate is 20 to 30%. And then in 1738, they did a small study with the 445 people, and they had 4% fatality. Again, much less if you have not gotten your vaccine, right? So, and important is that in 1749, Jenner was born, and Jenner is credited as the father of vaccine, and he is basically learned that the milkmaids who had cowpox were immune to smallpox, and he decided that to test it on a small boy, and he gave the small boy this milkmaid's saw from the milkmaid to the small boy, and then he treated the boy with real smallpox and cowpox, and it protected the small boy. And the word vaccination was introduced by his friend Richard Dunning, and of course now we don't call it variolation, we just call it vaccination. Vaca stands for Latin, for cow in Latin. But of course, everybody thought he is crazy, and the church called him ungodly and repulsive, and the Royal Society rejected the paper and warned him not to publish. And Edward Jenner persevered. He used his own funding and published it in 1798. Of course, 1840, there was free vaccination to all infants in UK. So this is the beginning of mass vaccination. And of course, all we hear about these days in the news is vaccine, 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 correct? So let's start. So since Jenner, there are many more vaccines than the list here, and it's all discovered by different people. And after smallpox, it was rabies, tuberculosis, polio, measles, and so forth. So I'm going to just move forward to say what is a good vaccine. A good vaccine should induce immune response without the disease. In other words, it should produce, your body should produce antibodies to neutralize. As soon as the pathogen or the virus or the bacteria enters, and it should neutralize the incoming bug, for example. And it should be non-toxic, shouldn't kill the patient. And we never know any toxins that actually kills patient. It also should be given, should give us lifelong immunity. And remember, many of the vaccines came into being, most of the vaccines happened only after 1920s. So people who were born then and later are just about getting old or gotten older, And we learned that the early childhood vaccine is not lifelong. So you got to give a booster vaccine to all the adults, the older adults. And now CDC, or Center for Disease Control in the US, as a whole entire adult vaccination schedule. And I highly recommend all ask your parents to get some of the critical vaccines, such as Shingrix against the shingles, which they would suffer as they age. So what's a good vaccine is something on the surface of the bacteria or surface of the virus that our immune system sees first, and this should be a good immunogen. That means it's good at getting the body to respond or produce antibodies, and it's usually a protein molecule. Okay, and then sometimes we do use polysaccharides for a specific bacteria, and these polysaccharides usually they're not a good immunogens, but if you conjugate it with something else, they do make a good vaccines. And then you can also create a vaccine against something produced by the pathogens, such as toxins. We can have, and toxins are essentially proteins. So, very brief overview of the vaccine immune response, because we don't have time to go into detail. When you get an injection of your vaccine, this is your skin, and the first thing happens is your innate immune system. So we have two major immune systems. One is the innate, that can see anything and it's not very specific. And then we have this adaptive immune system, and I'll come to it in a second. So in the innate immune system, as soon as we introduce either a virus or a protein, and this is taken up by dendritic cells and macrophages. And these are cells which are the soldiers. They are the first line soldiers. They are walking around looking to see if there's anybody who shouldn't be there has showed up. And they obviously know that when we introduce a vaccine, it's not normal, it's not part of human body, they immediately will engulf this vaccine or these proteins, and then they will travel to our lymphatic systems, the lymph nodes. Our body is full of lymph nodes, and in the lymph nodes they will recruit other soldiers, and now they just recruiting everybody they could. And the two main soldiers we deal with are the T cells and B cells, and the T cells will get activated, and these T cells will produce interferon gamma, which is an important cytokine. It will then cause the T cells to differentiate into cytotoxic T cells. Why is this T cells important? This is our next round of soldiers. They go and kill the host cells that are infected, or they will kill it's mainly to kill host cells that are infected, either by a virus or by a parasite, and so forth. Right? They also, in the lymph node, they will activate the B cells. What are B cells? B cells are they will differentiate into plasma cells. Plasma cells are the cells, is the factory that makes all the antibodies we need to fight any infections. So we need the plasma cells, but more importantly, in the lymph node, they also make what we call the memory B cells and memory T cells. These are the cells that will remember this vaccine or this virus or this pathogen. So the next time when your innate immune system sees them in the system, these memory B cells and T cells get activated. They don't have to go through this process directly. These memory B cells will differentiate into plasma cells and start making the antibody right away. So if you know immunology a little bit, what happens is people say, why do we need to get 2 shots, right? The reason we get 2 shots of the vaccine is, or even our body, when we come contact for the first time with the immunogen or an antigen, there's a very small or short primary immune response. That is the initial exposure. Again, this we talk about antibodies in general. There are 5 different types of antibodies, but for the primary immune response, we get what we call IgM. Okay, this shows up in 10 to 14 days, and then if you remember, all the vaccines have a 3 weeks period for a second shot, because when you come in with a second shot, what happens is the secondary immune response is much faster, and also this time they will generate IgG, and in addition they will generate your memory B cells and T cells. So this is the immune response 101, and hopefully this will make some sense later. So what are the various types of vaccine out there? So there are literally 5 kinds of vaccine, right? Initial vaccines were these live attenuated. In other words, you grow the strain over and over and over in a plate until it loses all its ability to cause disease, but still can grow and replicate in our body. So examples of attenuated viruses is polio, rota, measles, mumps, rubella. This is varicella zoster, that's the chicken pox, and yellow fever. And if you are pregnant, or you're trying to get pregnant, you are given the MMR vaccine, which is measles, mumps, rubella. They all are live attenuated vaccine. And then there's also attenuated bacteria. The most famous one is the BCG vaccine. That is basically Mycobacterium tuberculosis that has been passaged since 1930s. So it's a very old bacteria that we keep passaging it. And of course, the other one is the typhoid. The inactivated vaccine, okay, live attenuated, potentially it could give a little bit of disease. Some people feel more sick. So what they did is they started boiling the bacteria or boiling the virus, and then give that as the vaccine. Remember, vaccine is usually the protein. It doesn't matter it is a natural form or denatured form. The host cell will see it as a foreign, and it will generate antibody. So example of inactivated vaccine, the whole virus inactivation, polio, rabies, hepatitis A, flu, and of course we have SARS-CoV-2. And then the whole bacteria, where they also inactivated the whole bacteria. We have the Bordetella pertussis for whooping cough, and cholera vaccine for Vibrio cholera. And subunit vaccine is the best in the sense that you're not dealing with the whole cells, but they might have limited response, but many vaccines are subunit vaccine. That means part of a protein that's unique to that particular bacteria or that particular virus. And you can see we have hepatitis B, HPV, and so forth, and as well as shingles. Shingles, as I was telling you, Shingrix is for shingles, and that's a subunit vaccine. And then toxoids. What are toxoids? Inactivated toxins, right? So those are toxoids. And the polysaccharide vaccine, the most famous one is the Streptococcus pneumonia to prevent meningitis. In fact, all 3 polysaccharide vaccines are given to children to prevent meningitis. And then comes 2020, and then we have these new kinds of vaccines. This is vectored and messenger RNA. And the vectored vaccine is basically a DNA vaccine, where we introduce the DNA with the information we want into a particular virus that doesn't cause you a disease, and introduce it to human, and so that the virus will infect the human, but instead of causing a disease, it will introduce your antigen or your vaccine into the system. There was an emergency approval for Ebola in December 2019, and of course we have several recombinant vector or vaccine for SARS-CoV-2, and the most newest and most that gotten most attention, and for all good reasons, is the messenger RNA vaccine, and for the first time SARS-CoV-2 was introduced. But remember, all this vaccine based on DNA is not something happened this year. People have been working on it for 20 years or so on the DNA vaccine. But the idea to use messenger RNA as a vaccine was new, right? But all the tools were already there. It just needed somebody to have the vision to go to the next level. So once you have a vaccine, now you figure out vaccine, and you need to have an understanding what does it take from to go to the bench to population, right? So first you need to discover. Before you discovering, of course, you need to have a disease, and there is a reason that the countries are willing to spend money to understand the disease, and then from there you go into discovery, looking for a right vaccine or right molecule, and once it get passed through that phase, and you have a preclinical testing. Preclinical testing are things done in animals, in the lab, in the cell cultures, and then once that preclinical testing is positive, then you start the clinical research. This is where the human trials start. And I'm going to explain to you the various components of the human trial. And if this human trial, if the research is positive, and then they will submit it to Food and Drug Administration in the United States, and of course different countries have a different acronym for it. And here what happens is the data get analyzed. Even though the companies will submit a data that is analyzed, FDA want the raw data, because FDA will reanalyze the raw data and see that if their finding agrees with what the companies are submitting. And once FDA approves, then they give the company license to make more vaccine and do a next phase of studies, and then only then they go into large scale production. So it's a very lengthy process, and usually the usual timeline, this many years here is exploratory. You're learning the disease, learning about what can be done, and then the proof of concept, that's all preclinical, usually take 2 to 4 years, and then you have the clinical development, another 6 to 8 years, and then you register your vaccine, and you also have you file for the vaccine, and then you launch, and then you continue monitoring, a long-term monitoring. And this usually happens over a period of 10 to 12 years, if not longer. What happened in the past? In the past is you can't do one and do other. You have to do sequentially. With the COVID, it was so bad. COVID was so bad. Everything started happening side by side. And that really shortened the time. People did not shortchange the development or the I shouldn't say people, but most reputable companies did not shortchange any of these steps. The only thing they did is they did simultaneously. So while they were doing laboratory tests, isolation of virus, and getting the sequence, and this discovery preclinical testing also shortened by the fact that we have the bioinformatic analysis available. Those days we didn't know how to sequence, we didn't know how to compare sequence, we did not know how to identify best immunogen, but bioinformatic field really made a difference in our current research and also in the vaccine development. In addition, high performance computing. You needed the high performance computing to understand and compare and find the target, the most useful target to develop vaccine. And at the same time, what happens is they started doing the laboratory tests, and simultaneously they started doing the animal tests, and they started doing the challenge experiment, which means that you infect the animal with the virus, and then you give the animal vaccine, and then you infect it with the virus and see if the animals can fight the infection. So these steps was so condensed, and it happened instead of 3-4 years, we are doing this in 3-4 months. Everything is happening in a very, very short period of time. Again, sequentially versus simultaneously. So once we figured out there is a potential vaccine, and this whole clinical research actually has 4 different phases. So the first phase, phase 1, is once they approve for human testing, they have to test it for safety. So you give the vaccine to healthy people and see if the healthy people do react to the vaccine or not. And they usually use about 20 to 80 participants. In phase 2, once they say, okay, it's healthy people are not getting sick from the vaccine, so let's figure out how much do we need. Okay, so this time they also include patients and healthy people. So they'll figure out how safe it is with the patients, as well how much should we give to these patients to get a good immune response. And then even when that is good, then they say, okay, let's do a double-blind study. In other words, we recruit a lot more people, 300 to 3,000 people, and we give it divide the patients groups into two. We don't tell them what they are getting. So one group is getting a placebo, one group is getting vaccine. It's a double blind because the doctor doesn't know what he or she is giving to the patient, and the patient doesn't know what or she is receiving. So it is a completely blinded study, and once the study results comes out and you submit it for the approval. And here's where we need biostatisticians. They are extremely important part of this enterprise. They will go over this data and do the number crunching, and then they say, yes, this vaccine looks good, this is all the various outcome results, and etc. And of course the FDA says then we can go do phase 4. And now phase 4, you employ many participants, minimally 1,000 if not more, and you do this for number of years. And what is the difference now? Until now, we haven't entered phase 4 with all these vaccines, the SARS-CoV-2 vaccines, because the situation is so bad. FDA did an emergency use approval. That means this is an emergency, so we're going to go use it with just phase 3 data. All right, so far so good. And if there's no questions, I want to get into a little bit more into the virus now. Are we doing good?
Good, Prof.
Okay, excellent. So this is for people who are not in science, and some I see some people here who are friends who are here. So just some very brief background, right? So chromosomes are genetic materials, and they're made up of DNA. And there are 23 pairs of chromosomes we all carry. And just to give an idea, and each chromosome is between about, the smallest chromosome is about 1.53 centimeter. If you don't know what it is the Y chromosome. And the longest is 8.14 centimeter. There's a chromosome number 1. And this very small, but if you take all the chromosomes in our human body and attach them next to each other, they are about 6.5 feet long. Okay, and however they are packed so compact, so tight, you cannot see them. Right? They are very tiny. And so when you stretch this chromosome until to the point where you can actually see what it is made up of, it's made up of many bases. There are 4 kinds of bases, GATC, and the human genome contains 3 billion bases. So this is deoxyribonucleic acids, or DNA. So the DNA is, like I said, it's a genetic material, and it has to be transcribed to RNA, and this is our messenger, because proteins cannot be made of the DNA, but they can be made of the RNA. And when this RNA gets translated, they make the proteins that makes of our body, our hair, our nail, everything that happens in our body, we need the proteins. So this is the process going from DNA to RNA to protein. The process itself has a name, transcription, translation, and this is where you get the protein. But even though most genetic materials are DNA, there are few exceptions. Of course, exceptions are always hard to prove, and it took people a lot of time to prove there are exceptions, and they all are turned out to be viruses. And the RNA genome, example of viruses with RNA genome is your flu virus, right? and of course your SARS-CoV-2 virus, right? And SARS-CoV-2 has about 29.7 K bases. But what is the consequence of having RNA genome? Imagine you have DNA. A small change in one of these bases can cause a mutation. You may lose a limb. You may have you have a different color eye, or your hands doesn't form. It can have lots of consequences, right? So because of that, the process of passing your DNA from the mother to child, from the parents to the child, is fairly very faithful. It's faithfully copied and passed on as it replicates. And one of the reasons is the enzyme that replicates the DNA is highly faithful. However, the consequence of having RNA genome is that they are not faithful. RNA makes more RNA. The protein that makes more RNA, which is the RNA polymerase, it doesn't know how to not make mistakes. It makes so many mistakes, so the virus will keep mutating. So we'll come back to that later. I do want to have this slide to show the difference between three things, right? When we talk about SARS-CoV-2, we are talking about the virus. When we talk about COVID-19, that's coronavirus infectious diseases, we are talking about the disease. When we make vaccine is against the virus, not against the disease. So the vaccine is targeting the virus and virus particles, and the disease is what we have here. The common symptoms is dry cough, fatigue, fever, muscle ache, and there are a lot of uncommon symptoms: headache, loss of taste, sore throat, skin rash, vomiting, chills, discoloring and the extremities, and diarrhea. And then, of course, in the cases of severe symptoms, you have shortness of breath, difficulty breathing, you cough up blood, persistent chest pain and pressure, kidney failure, loss of white blood cells, as well as ultimately you have a heart attack. Rather, it's a pulmonary failure and cardiac failure. That is what happens when the severe cases of COVID, and when we could not do anything to the patient, that's what happens. So eventually we lose the patients. So the virus itself, and I told you already, it's an RNA genome. And if you look at the outside of the virus, it has this red color, what we call patterns of almost like a flowers, but these are the spike proteins, and this looks like the crown of like a crown. That's why they're known as the coronavirus. And what are these little red things? These are the things that makes the first contact to the host cell. When the virus enters our system, it has to find a way to enter the cell so that they can replicate. Remember, virus has to use the host system in order to replicate. And here's the spike protein, and you can see the protein that it binds to is ACE2 receptor, and you know that it uses two other receptors as well, but the most important thing is the ACE2 receptor. So the idea is, if I prevent this spike protein from binding to the host cell, then I would have prevented the infection, correct? So that is the whole premises for looking for a vaccine, and almost all the vaccines we have targets this protein called spike protein, which is found all over the virus, outside the virus. And as of 2020, we had about 184 vaccines in development. All kinds of active nuclear DNA, inactivated virus, live attenuated, and protein subunit. And as of May 2021, that is as of last week, 131 entered human trials. 4 already were abandoned after human trials. And here is the vaccines at the different phases. As you can see, at the moment there are 8 approved for full use, but really for mostly for emergency use, and there are several of them in the pipeline. The leading, here's the list of the leading vaccines. I'm highlighting the 5 vaccines that have been approved for in Malaysia. And we'll go through a little bit more in details. And these are the 5 vaccines that have been approved in Malaysia. The Pfizer BioNTech, and Pfizer BioNTech is a messenger RNA vaccine. They completed the phase 2 trial. Sinovac is an inactivated vaccine. It also has a phase 3 trial. I haven't seen the data. They're supposed to have done some clinical trials in Malaysia. I don't know what is the outcome. And then there are 3 vaccines that are adenovirus vaccine, where the DNA vaccine put in this adenovirus. They all are in the phase 2, 3, phase 3, phase 3 trials. We got this the data from phase 2, 3 from AstraZeneca. We don't have much information on both the Chinese or this is the Russian Sputnik 5. This is a Russian vaccine that also we don't have much information, but they all are target except for the inactivated vaccine, which is the Sinovac. The others all target the spike protein. And then the mRNA vaccine is a very special vaccine, and I have a small video to show you, and I should have had this ready, but give me a second. Hopefully it will have less of a problem today. Can you guys see the video? Let's hope you can hear it too. Can you hear?
Yeah.
Can you hear?
Not yet. No.
Hang on. Why can't you hear? All right, so let me try that. Can you hear?
Not yet. Not yet.
Not yet? Hang on, guys.
Yeah. Share stop. Okay.
Stop share first.
Stop share, and when you share again, and then share the sound. That's a small box.
I see. You can hear me still, right?
Yeah, I can hear you.
Okay, so share screen.
And then share sound.
Is it working?
No, when you share screen, there's a small box there, share sound.
Share screen.
Stop it. Share screen.
Hang on, sorry.
There is, yeah.
Share sound, yes. Thank you.
Yeah.
All right, so can you hear?
All started with isolation.
Now you can?
Perfect.
It all started with isolation of the virus from an infected patient in China. The coronavirus was sequenced and its genetic information was uploaded to a public database. Working together, scientists at the NIH and Moderna identified the sequence for a key protein on the surface of the virus, called the spike protein, as a good vaccine candidate. The instructions for making the spike protein were then encoded into an instruction molecule called mRNA, which could be administered directly to patients as a vaccine. The vaccine is injected just like other vaccines. The mRNA is taken to immune cells, where it instructs cells to make copies of the spike protein as if the cells had been infected by the coronavirus. Other immune cells, shown here as red and orange figures, are then able to learn about the spike protein and develop ways to protect the person if they ever come into contact with the actual coronavirus. What makes this approach different is that you don't need to make the virus itself to make a vaccine, a time-consuming and intensive process. Instead, you use the information from the virus and administer the information directly to the patient. Essentially, the patient makes their own vaccine. This cuts out the middleman. Using this approach, NIH and Moderna were able to go from sequence to a vaccine ready for human testing in record time, just 42 days.
Okay, so even though this is Moderna, but it's the same technology, right? Why am I not? Okay, share screen again, back to my slides. So I hope that video was informative. It's a simple, but it's a very nice video. I thought that I don't have to spend time explaining how RNA vaccine works. So what is important is when you use a protein as a vaccine, the purification step is so onerous. It's so much work, and to get it pure, to get it clean, and this way we just have to purify an RNA, and you put it in a little lipid molecule and introduce to the human cells into our body, and our body makes its own vaccine. How about the viral vaccines or the DNA vaccines? You take the DNA that codes for your spike protein, put it inside a virus, and then put it in this special virus that then will infect will enter our cells, and then will start making our body again, telling our body to make the protein we need. And a simpler figure is right here. So the virus that everybody is using is the adenovirus. It causes a little mild flu-like symptoms, and then you put the piece of DNA in the virus head, and doesn't have anything else. It's just this capsid. We call this a viral head or the capsid, and this virus attaches to the cell. It enters the cell, and this virus will only release the DNA when it gets to the nucleus. So it makes its way to the nucleus and injects the DNA inside the cell, which then makes the host will make the messenger RNA, and then messenger RNA will come out into the cytoplasm, and then it will make all the spike protein we need. And now this cell will have the spike protein on the surface, which is then get recognized by our immune system as this is no longer a good cell that we need to kill these cells, and eventually they make antibodies to this spike protein, because the host cell will recognize this spike protein on these cells as a foreign entity. So once they decide, I'm just not going to go into detail. What I want to tell you is that, when they apply for approval, they have to disclose everything. They have to disclose how it is made, what it contains, what are other chemicals that contains. And as a result, we know a lot about Pfizer BioNTech and AstraZeneca and Moderna vaccine. What we don't know much about is Sputnik 5, CanSino, and Sinovac. So these are the 5 vaccines that have been approved for Malaysia. We do not have enough information. And we also know that many of these vaccines, the temperature is an important. How do you keep them, how stable they are? And now we know that Pfizer started out needing minus 70 degrees. Now they have dropped it to minus 20 degrees. Regardless, Pfizer is a little bit fragile compared to all the other vaccine that can be kept at 2 to 8 degrees. And then in terms of delivery, how are they delivered? Of course, the messenger RNA vaccines are delivered in a lipid nanoparticles. We know exactly what it has. And once again, I cannot emphasize, I don't know how they are, what else is in these other vaccines, but they all are given as intramuscular injections, and they're all given as 2 doses, and depends on the vaccine, either needed 22 days or 28 days. So this is how did they come up with these dates. They just decided they will do the experiments with these dates when they did the human trials. So we are going to go with the human trial dates. And then in terms of phase 2 and 3 clinical trials, and I put this slide up just to tell you that again we lack a lot of information for Sputnik, Sinovac, and CanSino, whereas we know a lot about the other vaccines. We know the sites they used for clinical trials. Pfizer probably had the most number of countries, so they used 3, 4, 6 countries where they did the human trial. AstraZeneca did in 4 countries, and Sputnik all their human trials in Russia, and Sinovac, this is not US, sorry, this is China, in both cases China. And the other important thing also to notice is that most of the human trials that people use that, they have, most of the time there are more males and females, and as you can see, Sputnik had 60% more males, and then AstraZeneca had slightly more females, and then Pfizer is equal amount. So there's always that gender difference because male and female react to it differently. Another big issue with all these vaccine human trials is the fact that most of the participants in the human trials are mostly whites, or the Chinese in China case, in Russia is all Russians. And most of it, as you can see based on the numbers here, Pfizer had 82% whites, Sputnik at almost 198.5% whites, and then AstraZeneca had also 82% white. And then the age also varied all over. What I'm trying to get to tell you is that we don't know a whole lot about how these vaccines work for Malays, the Indians. Of course, the Chinese, because of the China vaccine with their experiments with Chinese, that might still very good, right? And then in terms of what kind of outcome measures did they analyze? What did they look at? Did they monitor the patients? When did they monitor those people? And what are the various aspects of the vaccine did they look at? And again, I have to tell you that I only have some information for these three vaccines, and they all looked at how whether they get infected, there was any adverse reactions, there was side effects, and in some cases they want to check if the antibody is there is a vaccine specific antibody in your body, whether it can neutralize the viruses. And then in terms of side effects, Pfizer and AstraZeneca reported a long list of side effects, and they monitored all these. And for Sputnik, I know we only know that people get headache and fever. We don't know anything else. And for CoronaVac and CanSino, we don't know what other side effects. And I also want to show you this slide. And somebody asked if I have a choice what vaccine I will take, and of course I will take Moderna. However, I only had access to Pfizer, so I've taken my Pfizer vaccine. That's because the Pfizer vaccine had a little bit more side effects as compared to Moderna, and you can see. And the most of the side effects were general reaction at the injection site. Side effects, nothing major. And for Sputnik and this Chinese vaccines, I don't know what is the outcome. So we basically talked about the vaccine efficacy, and I wanted to tell you that the number of agents used in these studies were very low or none. So if they say that it's 95% efficacious, then it is 95% efficacious in the white population rather than the Asian population. So here, coming back to the 5 vaccines in Malaysia. And so the Pfizer is, as you know, has several different names, and it is your mRNA vaccine. The AstraZeneca is your Vaxvaria, Covishield, the COVAX vaccine in Malaysia, and then the Sputnik 5 is also known as Gum COVID Vac, and then CanSino Bio is Convidecia, and Sinovac is CoronaVac, and what else? The other thing that look at this percentage efficacy. Pfizer has 91, AstraZeneca 76. What does that mean? If I say 70, let's say this vaccine has 80% efficacious, that means 2 out of 10 people will not respond to this vaccine. Okay, so 2 out of 10 people have 0 response to this vaccine, and that's what you should look at. And you can see Sinovac had 2 different human trials in Brazil. It was only 51% efficacious. That means every 1 in 2% this vaccine would work. So what does that mean to us? It's a big open question. Whereas in Turkey, where most of them, it's a mixed population, and they had about 91% efficacy for this Sinovac vaccine. So, I don't know if you have a choice what you should choose. But if you don't have a choice, take whatever comes your way, because having vaccine outdoes not having a vaccine. And in terms of what has Malaysia done, Malaysia has gotten 32 million doses of from Pfizer. And that is literally 1 dose for every person in the community. And then AstraZeneca is 12.8 million, and then Sputnik, they gotten 6.4 million doses. CanSino Bio 3.5 million, and Sinovac, they have 12 million doses. So you will have access to those vaccines. So the other important thing that the on Malaysian website, it says this vaccine has been approved many countries, right? And I think that's a misinformation. So you should really look at that information carefully, because for example, Pfizer BioNTech is only been approved in Brazil and Saudi Arabia. All the other countries is only for emergency use. In other words, they think it's important, they should use it. But approval is different from emergency use. And you can see, sorry, New Zealand, Brazil, and Saudi Arabia is where Pfizer BioNTech has been approved. Canada, US, and all this is just emergency use, including Malaysia right here. And Oxford AstraZeneca, again, only approved for Brazil. We still haven't approved for US, not even for emergency use. And you can see the number of countries that include many of the South Asian countries that have been approved. Sputnik 5 is only, it's only early use for even to Russia. They have not approved it for complete use. They only use for early and emergency use, and that includes India, and Malaysia didn't make the map yet here. And then CanSino, and you can see it's been approved in China. You have only 4 countries that have been approved for early use: Mexico, Chile, Pakistan, and Hungary. And none of the other countries have even accepted CanSino yet. And finally, Sinovac, approved for China, as you can imagine, and few of these countries for early or limited use. So in other words, not the data is given, you got to take it, and you should do your homework to find out, is it really approved in all over the world? No, only for limited use. However, all cases of vaccine, and I have data for 3. One is the fact that the people who get the vaccine have less severe COVID. Here's with Pfizer. Here's the vaccine group. They had only 1 case of COVID, and this is the group that did not get any vaccine. We had severe COVID all along the graph here. The same thing, they got Sputnik. These people who got vaccine did not get COVID, and this is the group of patients who got placebo. They ended up getting vaccine, COVID. Right? But these are severe COVID that the people ended up dying. Similar results for AstraZeneca vaccine also had the similar results. In other words, all vaccine will work on reducing severity and protect you from the deaths, right? Protect from the patient from dying. So obviously vaccine is extremely useful. If you don't believe it, here's another example. It's not a SARS-CoV-2 vaccine, it's a flu vaccine. I preach that everybody should get the flu vaccine. As I told you, flu mutates. It's an RNA virus. If you don't take the vaccine, in fact, between 2018 and 19, this is a US record, there were 35.5 million people could have prevented. It could have prevented 65 million from medical visits, about 500,000 of hospitalization. Importantly, it could have prevented 34,000 people dying every year. This is every year ever since flu has been around, because people don't take vaccine. Of course, SARS-CoV-2, that toll is much higher. And the other reason to take vaccine, because of without the COVID vaccine, without the vaccination, and with all the shutdowns and all this economic crisis, more importantly, about 88 to 115 million people will slip into extreme poverty. In other words, extreme poverty is defined as the person having only 5 ringgit per day to spend, right? So if you take about 30 days, right? So you only have 5 ringgit per day to spend on everything you need, your food, your water, anything, all your essentials. So if you don't have vaccine and if you stop taking vaccine, this only will get worse. And this is and now not only COVID is causing economic crisis, but there are going to be millions of people who become very, very poor. And you can only imagine what kind of political upheaval that will lead to. Right? And though vaccines had so much good, so much protection that can help, however, there weren't many vaccines available. There are many vaccine preventable diseases. And one of the reasons we didn't have many vaccines is because, remember, it takes you 10 years, 12 years to make. It will also cost more than $1 billion to develop a vaccine. And so the companies rather give you the medication to continue treating you rather than giving you a vaccine to cure you for lifetime. So they weren't interested in spending the time and energy to develop vaccine. But why suddenly all these companies are making the vaccine, right? Why all of a sudden there's like 100 plus companies making vaccine? Because of the COVID burden. And this is January 28th. And that I downloaded, and you can see this high burden and growing, and that included Malaysia. This is in early January, and of course South America and Mexico and Europe and Libya. They were all Botswana and all these places. They were high burden and growing. What is the global outlook right now? Oh, sorry. Before I go there. The other thing that happened, and this is today's numbers, the number of people who are dying was so high that all the companies thought that they have to do something. But more importantly, the government said we have to do something because they decided to give them money before they developed the vaccine. Because we have about 158 million cases, and of these 3.2 million have died. And here is the United States with the most number of deaths. Okay, this is more than 500,000, and here is Malaysia with 1,700 as of today. Right? This is today's number. So there are more people dying in the richer countries, and as a result, like US put in I don't know how many billions, $5 to $10 billion on vaccine development, and they gave the company the money upfront so they can start working on the vaccine on all levels at the same time. But don't become only 1,700. What happens in that 1,700 is your entire family is included. So not even one death is acceptable. Okay, the number may be small, but it's not acceptable. And in fact, this is May 9th. That is literally 2 days ago. And the countries where the vaccine is high burden and growing. Can you see where Malaysia is? And that's India. India, Malaysia, Georgia, Netherlands, Denmark, Botswana, and then there's Costa Rica, Ecuador, Bolivia, and Suriname and Brazil. These are, and then Panama is right here, okay? These are the countries where the burden is high and it is growing. It's not slow down yet. So Malaysia is in that list of countries and it's a huge concern, and as you can imagine, it's a huge concern for me too, having all my loved ones in Malaysia as we speak. And so if you look at the numbers as of today, we had 70 new deaths. We have total of 1,700 deaths. We have almost 500,000 of confirmed cases. On May 3rd, we had 136 people dying. And then what about, so we are about 33 million in population, and as of May 3rd, there's only 1.4 million that has been vaccinated. That is a shame. And here you can see, people became very complacent. Oh, the virus is only affecting US. We are fine. So the number came down, and then we all decided, okay, it's not going to come to Malaysia. And you went out without your mask and had all your partying and whatever not. And the death started going up. It came down a little bit, and now it's on the way up. And you all each and every one of you who are in Malaysia right now have to be concerned. This 136 may be your parents, your grandparents, your aunts and uncles, your brothers and sisters. So we must do everything to stop this, which means get vaccinated, making sure our family members get vaccinated, and making sure you mask up, stop going out and doing things that you shouldn't be doing. So I put here an abysmal statistics. As of today, there's only 10 million people. That's only 1/3 of the population have registered. Here's the registration link, right, to get the vaccine. And only about 1.1 million people gotten vaccinated, and Sabah has better numbers, and you can see Perak and Kedah, where you guys are. It's a shame. Penang is doing well, congratulations, but not well enough, right? And the rest of the country is not doing too well. So we must do everything. And if you want to wait for everybody to get vaccinated for herd immunity, you need to get 80% vaccinated. So we are not anywhere close, which means that US is not going to let me visit Malaysia for a while to come. And if you look state-wise, Kedah is doing terrible. Right, there's only 500,000 registered, only that's about 33%, and Perak is also terrible, where I come from, and 37% of the population have registered, not gotten the vaccine yet. And okay, and in terms of vaccination, that's even worse. Kedah has only 4.4% and Perak has 5%. The only place that has highest number of vaccination, I believe, is Putrajaya. Maybe they had no choice, right? If you want to come to work, you better get vaccinated. So this was updated as of this morning, and you can see that for you guys 2 days ago. It's very sad to see these numbers. And some of you say, hey, what's the point? Why should I get vaccinated, right? Because it's mutating and I shouldn't bother. And so I'm going to very quickly tell you, yes, the virus is mutating. I already told you why it is mutating. And here are the various mutants. In fact, they have been sequencing the entire genome, and you can see there are many mutants that have appeared. And this just started in 19th December, and here's 21st April. And there are many variants. And the most important variants are the 6 variants: UK, South Africa, Brazil, California, New York, and Indian variant. And what about these variants? We know that the UK variant, Pfizer, AstraZeneca, and Novavax vaccine works. We have proof based on clinical trials and lab and real-world data. The South African variant, depending on the vaccine, it is works or not. There's a variable. Brazil is also variable efficacy. How about for this California, New York, and Indian variants? That we only have lab results. We don't know if the vaccines works, but in the lab, the vaccines can neutralize these various variants. So that's a big question mark. So what I hope I've convinced you is that the vaccine wasn't rushed. It's mostly safe. The body cannot cope without vaccine, because if it can cope, people won't be dying. Vaccine is just a part of the protein. It will never give you COVID. And they will believe that in the vaccine there were chips. That's all, crazy talk. It will not tweak your DNA. Side effects are worse than COVID-19? I don't think so. Is dying is worse than living? No. Make women sterile? No proof. Cause autism and diabetes? There's never proof that any vaccine caused autism or diabetes. Cannot protect from variants. All I can say is that it can protect from most of the variants we know. We don't have enough clinical data to support it. So vaccine is safe and effective. It can prevent deadly illness. It can better immunity than natural infections. If you do not get vaccinated, deaths will continue to increase, and you at the end you will be a nameless person being buried. Here's an example of China. This is in New York, and of course this is in India. There are bodies getting ready to be burned, and the choice is yours. right? If you do get COVID, you can't be next to your loved ones, cannot be next to you. So you always have to consider the efficacy versus risk, right? You know the risk, you know the efficacy, and ultimately the question to ask to live or not to live. I choose to live, so I will take the risk and get the vaccine. So with that, I thank you, and I'll stop here.
Thank you, Prof. Kalai. You have a lot of energy. And we have a good number of audience here watching. We have around 78 participants in our Zoom link, and also we have a YouTube streaming is going on. I hope somebody is watching the questions from YouTube. And I think Prof. Kalai, you covered from the fundamental immunology to various types of vaccine, how the macrophage and dendritic cell talk to each other, and different types of vaccine. I think it is very, very useful, at least for everyone who's watching, to know the foundation. Particularly, you covered about the polio vaccine, and that is very interesting. And also, you thought about the different phases of clinical trial and different types of corona vaccines, and you gave judgment about the types of vaccine Malaysia is planning to embark on, and your in-depth analysis is very much appreciated. The vaccine coverage, I think that is the end of the day. To end the game, I think we need to have the vaccine coverage, and you gave your wisdom on it, the balance between having a long COVID and escaping with the vaccine. I think that is the thing. We really appreciate all your input and very frank opinion. I think we have so many questions that are coming from different direction, and I hope you also have a team on your side to get your questions, and we will try.
Can somebody post the question? Can somebody summarize? Give me one or two questions here.
Actually, this early morning also I received some question from the question.
Oh, there was, yes, you did send me. So somebody died immediately after vaccine. That must be a fluke case. I don't think there is no evidence to say anybody would die from getting a vaccine unless this person had some major underlying issues. So that's I would not even propagate that information.
Just a mere coincidence, bro?
Yeah, that's a mere coincidence. Yes, yeah. And the other question is, do we need a booster down the road? And the answer is yes. I really believe that we all would need a booster. And so as a result, I think the Pfizer, the companies that do the mRNA vaccine in particularly, they're going to be in business for a very long time. Because the advantage of the mRNA vaccine is to change the mRNA every day. They can make that vaccine to suit for that particular variant to that particular country in a very quickly. So yes, and I would, especially now we know 2-3 months later, even sometimes a little bit later, the antibodies are waning, but we don't want to get infected to find out, but we certainly would be needing booster vaccines.
Yeah. And the other question was about, can this vaccine lead to a severe gene mutation? Somebody asked.
No, how would it? So now that you know the science behind, I hope I did cover the science. Yeah. How can you imagine that the vaccine will make your gene mutate? And also remember, this is an RNA virus. What does that mean? This virus never enter our nucleus. So it's going to replicate in the cytoplasm. The virus itself never makes to the nucleus where your DNA resides. How can it do anything to the DNA? And then when we do send the DNA vaccine with using the adenovirus, it only takes the viral DNA, nothing else, no enzymes, no proteins. The DNA gets injected inside the cell into the nucleus, and the host machinery that transcribes and translates. So there is no way, not even remotely possible for your DNA to mutate.
Okay, that's very good. Thank you, Prof. And one question was about the long-term effect of the vaccine. What do you think?
I don't think the long-term effect, I don't think there is any long-term effect, but there is the long-term effect of COVID, right? What we call the long haulers. People are having issues. And I want to liken that to if you have a strep infection, right? If you have a strep infection and if you don't treat it in time, and you might get arthritis, or rheumatic arthritis. And that is because our body makes antibodies to the bacterial protein, which then recognizes our muscles and joints, and then it makes autoantibodies. So that may be an issue with post infection to have some long term effect, but not vaccine.
Okay. And there's one question about the blood clot and the vaccine.
Very good question. It's a very big, long answer. And actually, I do have a slide if you don't mind me sharing. I have lots of slides that I could share. So, you see this slide?
Yeah.
Right, so this slide is, it really gets into COVID itself, right? And when you have the viral infection, you'll be asymptomatic, no symptoms, but you're PCR plus, and then you have a fever, a dry cough early, and then there is this shortness of breath. That's your stage 2, and hypoxia, and then you have this hyperinflammatory phase where you have all the problems. And here is the very detailed, this is a work that we published late last year. And when the virus attacks our cells, there are many things that go wrong. But one of the things that happens is thrombosis. Because this is because of the inflammation, the cells are being attacked by our system. You have a lot of tissue damage, so you will have a lot of blood clotting to prevent, and then the host immune cells coming and they also breaking up and create this net, and all that causes a lot of thrombosis. So this could be a reason why that we do have blood clots in the process, because of so much tissue damage. And our body then will have to remember, it is constantly have to replenish. You have a tissue damage, it go fixes it, and the virus attacks, and then you keep fixing it. In the process, you'll end up clotting, and the clots have to be removed. It looks like the virus itself has ways to hijack that system, that whole cycle of replenishing and removing the clots. So that's the reason that COVID infections will itself have clot. And then in terms of AstraZeneca, I believe that had after vaccine people had thrombosis, right? I think that's a concern that people had. And that's, there are many reasons, and it may be because there are some autoantibodies in the system, but the answer is not very clear. The first few cases of thrombosis in the AstraZeneca case were not at all associated with the vaccine itself. But having said that, I think the thrombosis in the COVID patients chances are higher to have thrombosis in the COVID patients than due to vaccine.
There are so many questions here.
Please, I have all the time if you have the time.
I have to sleep late a little bit today. So there was one question from, actually the questions all are running up. I'm going crazy reading it. Okay, there's one question from Ms. Shyamala Devi. She's asking that if there's any adverse effect on elderly cancer patients receiving chemotherapy and radiotherapy. Means maybe they're trying to ask, they can take, cancer patient, can they take the vaccine while doing the chemotherapy and radiotherapy?
So I don't know the right answer. They should really talk to their doctor, and in fact, actually go to American Cancer Society website, and they have a lot of instructions to who should get vaccine and when you should get vaccine. If you're going for chemotherapy, when you should halt the chemotherapy. So all those details are on the website. American Cancer Society has a very good vaccine advisory. In fact, all the major disease societies in Alzheimer's disease and cystic fibrosis and everything, those society website has the first page is COVID advisory and vaccine advisory documents. So I would recommend familiarize with that information. So when you do talk to your doctor, you can print out and take it. Hey, look, this is what it says.
Okay, there is one smart questions, one again from Ms. Shamila. She says, should we take aspirin to prevent blood clots?
Well, they recommend most of the treatment we get is all supportive, right? So if it's a body ache and all that is supportive, and I don't see any reason why not. And again, I'm not a medical doctor. If you ask me, will I take a medicine to prevent my body ache? Yes, I will. But again, I will couch it with the fact that I'm not a medical doctor.
Yeah, same actually. You read the question, means the same similar questions. The one person is asking, should we take paracetamol just before we take the vaccine? As I think we explained it about it.
Well, you don't take paracetamol unnecessarily, right? So, and you should, if you're a person who takes a lot of this medication every day, you might want to check if there's any adverse effects. And I can tell you, somebody who takes, quite a few medications myself, I had no side effects.
Okay. I think the participant, if I miss any question, you can post it again.
Oh, they can ask too.
You can ask. You can open your mic and ask, please.
Yes.
Yeah. So while we are waiting for a question, Prof. Kalai, what do you think about the triple mutant they're talking about in India?
So the India variant right now, I was actually looking at it. So the Indian variant, we still, we don't know if the variant is how bad is this, whether it will not respond to the vaccine or not. I think the jury is still out. I think they have to do the experiments. And there's no time to do human experiments, so they have to do some laboratory experiments to show these current vaccines will work. And I can tell you that the P1 variant does not respond to the vaccine already. So.
Okay. Okay. There's one question from Kalaivani Kaliaperumal. She asked about, does black fungus infection has any effect on COVID? I think she's asking about mucormycosis in.
Yeah, I heard that. I have no idea. I don't know. That probably has an unrelated.
Yeah, I think due to the immunosuppression and other diabetes and other reasons. Right.
But remember, when you have COVID, your immune system is overactive, trying to get rid of the virus. So that, because of all the tissue damage, if you are exposed to any other fungi or any other viruses, particularly fungi, they can start growing on the tissue. Remember, the one thing to remember is the viruses cannot penetrate the first line of defense. Your first line of defense is your skin, then followed by your mucus. But with the COVID, the first line of defense is completely breached. Your mucus layer in your lungs is completely breached. So anything else in your lungs can cause a lot of issues. So it is what you call secondary infections. You cannot stop secondary infections, and it only can make matters worse for the virus as well, for the person to get sicker because of the virus and because of the secondary infection. What we don't know is that how many of these patients are dying from secondary infection. There are anecdotal discussion among medical in our profession that lot of these people with COVID has a high level of antibiotic resistant bacterial infections, but there's not enough data that have been collected because that is all as a function of the tissue damage due to COVID.
I think there are one more question related to the drug. For a cardio patient who is on aspirin, would it be safe for a Pfizer vaccine? I think again a drug-related question.
Go and look up the cardiovascular society page, right? There's a very detailed explanation for people with cardiovascular diseases, what kind of potential cross reaction. But I can tell you, to date, we don't have enough data to. There's not enough published information, so they all will give you educated guesses to what is good and bad. And again, I refer you to those particular links. Maybe Renu, the society, the student body can make a list of various links to people can go and cross-check. Make this a public service, and you can talk to me about it, where are all the cross places you can get this information. And a CDC, Center for Disease Control, USA, right, cdc.gov, and PHS England, and WHO, all three websites have a very extensive discussions and recommendations on the vaccines and who should and should not who should be excluded and included. So you should go look it up.
There are three more questions there. Let me.
I have zero reaction to my vaccine. Okay, you can ask Amrita. She thought I was kidding, but I think sometimes it's all mental. You got to go with the saying that you are going to be the one of the person who's not going to have any reaction. Remember, only 3% of the people with Pfizer had any kind of side effect, such as pain on the site of infection or redness, nothing more. And then there's also this herd effect of certain people falling ill, and then everybody thinks they are ill, and then what can you say?
There is one question from Christian Castellanos.
Castellanos, yes.
Yeah. So he's asking about the comparing vaccine efficiency between ethnic groups and asking what factor can affect the efficacy of a vaccine by ethnicity.
So Christian Castellanos is logging in from Bogota, Colombia. He's actually my student who's back home for summer. So yes, as you noticed very briefly, I didn't have a lot of time. Most of the vaccines are tested with mainly Caucasians, so what we call whites. And the difference is that our genetic makeup is different. Right, we all respond to infections differently. We all respond to and vaccines differently. So we do, and because our genetic makeup is so different, every vaccine should be tested. I don't know if you know that Japan doesn't let anybody bring any drugs into Japan until they retest all the experiments with the Japanese. Mm-hmm. But rest of the world will take the word for the company saying that it works in this population, so as a result it should work in every population. But that's not the case in Japan. In Japan, you can get all the countries will approve the drugs. The Japan will only approve it until the Japanese have done the test in Japan. Right? So we all, as our food, our genetic makeup is different, our food intake is different, our cultures, customs are different. So we all will react differently to the vaccines. We ought to.
I think I have to thank Dr. Girish has been answering some of the questions. And there's one nice question for you. How do Prof. Mati feels post-vaccination?
Nothing, as good as ever.
Yeah, I do had 2 doses. I'm also free. Nothing happened. We are still.
Yeah, nothing happened. Even after first dose or second dose, I didn't feel and I just felt a little prick, and that's it. Nothing more, nothing less, and life goes on. It's the same thing. People ask me, do I have what jet lag? I say I don't have time to think about jet lag. So if you sit and think about all the pains, you might have it all.
Okay, there is an interesting question. I think keep on asking. It's from Harish Acharya. And he's asking, I think it's asking about the receptor binding site, the spike receptor binding site, and the vaccine efficacy, mutation in the receptor binding site and the vaccine efficacy.
So the mutation is not in the receptor binding the mutation is in the spike protein, right? And the mutation is not limited to the spike protein. Remember, the RNA virus uses its own enzyme to replicate its genome, the RNA genome. So there are mutations all over. There is a website, I can put it there, where all the current mutations has become available, right? And most the mutations is not limited to, but just like any other, just give me 1 second, I will show you the link, because in fact I've got that image from there. Come on. Okay, so while I'm trying to find that link to you guys, so they have been, since December, they have been tracking all the mutations. So its mutations are all over the RNA genome, and unfortunately, the most mutations are on the spike protein, right? And the spike protein is because, just like when we give antibiotics to get rid of the bacteria, what does the bacteria do? It fights back for its survival. It becomes resistant. And same thing in this case, the virus has no intention of killing you because it needs to survive. But in the process, what kills you is your own immune system fighting the virus. So the virus then, and it the proportionately higher mutation seems to happen on the spike protein. There, and then if I can find it, I'll show you this link where we actually shows where all the mutations are.
Okay, you want me to share the picture I shared with you, all the mutations?
And I have a better one here. Let me
okay.
show you. Yes, so this is an amazing link, because yours shows all the mutations in all the things. So here is this website, right? It's they started looking at the you all can see the site.
No, now it's okay.
And now it's okay. And here is that figure I got with where the vaccine, where all the mutations are, and you can actually go to the website and look at different ways to look at how these mutations look. But the thing that I really like is this one. And here is this genome of the SARS-CoV-2 virus, and you can see the mutations are all over, right? This spike here, the bar here shows where the number of mutants are, and you can see this is the spike protein, and a lot of it is in the spike protein as compared to ORF1b, and then this one of the ORFs here, right here, this is also quite a few of these things, ORF8, and it's very interesting, the mutations are in ORF8 because ORF8 is also the ORF, the open reading frame, the protein that hijacks the plasmid a process of removing the clotting process. So our body must be fighting against that protein as well. So there is it's a battle between the virus and our immune system, and in the cases where the people die is that our immune system lost the battle. In the end it destroyed our lungs, and then people die not from COVID, but they die from the side effect of COVID, that is the respiratory failure and cardiac failure.
Yeah. Is there any more questions coming up? Okay, maybe one final questions. I think the whole world is now looking at why there is a steep increase in cases and why there is a second wave. Prof. Kala can summarize and educate the audience and make sure it doesn't happen in Malaysia.
Yeah, I am so concerned it's already happening in Malaysia. So in the case of India, and again, this is analyzing what's going on, and I will say that it does not represent my society or my institution, but this is personal professional opinion, is that the people became lackadaisical. In other words, people decided that the virus is in the US, it's not going to make it here. So we have managed to somehow dodge the bullet in the first 1 year, so we can go back to normal and just do not take the precautions. And in India, as you know, there was a Holi, and there was this big Kumbh Mela and all kinds of festival. And to compound it, they had elections where people came into the cities and went back, and all that's contributed to explosion of infection and these young people going back home and also taking the virus back home. So this is again, no, it's before then. So, and then during this process, of course, you also will have the variants that are now, mutate, and then they will also contribute to the problem. So in Malaysia, and we also becoming, I think right now as on a lockdown, I'm glad to hear that. But I think but more than the lockdown, we need to get everybody vaccinated. And that's only, you're never going to get rid of the virus, right? All you can do is to prevent severity of the disease and ability to fight back. And so that's, at some point the virus is just like, if you think about it, a very good example is the flu virus. 1918 flu killed 1/3 of the world population, a 1908 flu, right? But and then somehow the virus figured a way to live in equilibrium with the human population, so the sickness is milder, and then if you get the vaccine, you don't even get sick or even milder. So we'll have to live with coronavirus rest of our lifetime, and your children will. But how do we minimize the collateral damage from this particular infection? And it's vaccination.
We are entering a new era, but we still survive. I think we all are successful. And
Yeah.
I think, Prof, you rightly said that the variation mutants and lack of SOP and social distancing and vaccine hesitancy all together played the role in the second wave of COVID, what we are seeing now.
Yeah.
And
I hope AIMS has 100% vaccination policy.
we are Malaysian policies opt-in. So all our frontliners are vaccinated. I'm glad to say I'm the COVID task force chairman here, and I get non-stop call. Basically, can't sleep. All our frontliners are even final year medical students, all are vaccinated in the campus. So hopefully our dream is to get even our wardens all are vaccinated.
Excellent. Yeah.
And so that is status here. We hope, and our dream will be the once we cross the 80% vaccination in the whole country, I think we can dream of going around.
Okay, so I see one unrelated question
Okay.
that is nothing to do with vaccine. Chai Hui Chi is asking about how you can pursue research. Why don't you guys have a separate session that doesn't involve all these individuals in this call? We can talk about pursuing education abroad.
Yeah, ABSA can organize a separate session on it. Mr. Hugo is the president, can take note, and we can have a separate discussion on it.
Right, yeah.
One last question.
Any interesting questions, Giri?
Yeah, I can ask one last question, if you allow me. That's the mutation. You know, the mutations happening. Do we miss the diagnosis by RT-PCR?
So at the moment, if the mutation is happening at that the primers we are using, they should be constantly changing the primer. I don't think so, right? Because even if they know particular variant is there, changing the primers for RT-PCR is just like it's no brainer, right? So
Yeah.
you should constantly update the primers they use if they're looking for a specific variants.
Yes.
They shouldn't miss.
I think normally they look for more than one gene, if I'm not mistaken. So
I really don't know. I'm sure they probably use. If I were them looking at the mutation rate and from the graph, there are regions of the virus doesn't seems to have many mutations. So I would design primers that are specific for those regions of the virus that doesn't undergo mutation. So we don't know what primers the different companies are using, and I'm sure if I'm the company CEO, I would be literally a company scientist looking at that particular set of data and design primers that are universal for all SARS-CoV-2 virus.
we have companies in Malaysia. They manufacture both antigen detection kit and also RT-PCR based kits.
And that antigen detection kits it can miss because we already know the neutralizing antibodies of the vaccine don't recognize some of the variants. So if that antigen detection kits used a variant that is no longer around, that test can miss. And those tests are much hard. Those kits are a lot more harder. Like they have to, express the protein and purify the protein in large amount to create the ELISA plates, and so that's a bit more work than just changing the PCR primer, which is much cheaper and quicker. So I would, with the variants changing in such a high rate, I would trust the PCR test more than the antibody test.
Yes.
If it's negative, we don't know why it is negative. If it's positive, it's there.
I think it is already going to be 11 o'clock for you. And
Yes.
thank you so much, Prof. Kalai. I think I'll pass to thank you. On behalf of AIMS University, I think it is the first talk and it's a very impactful talk. And Prof, I also got a request from our collaborator in India. They are requesting you to give a talk. I think you will be kind enough to give talk to them also. I think many of them are really wanted to know fundamentals and basics about it. There are some college from Madurai they requested just now. I'll pass to you. And
Sure.
I hope that this is the way we can demystify them and then give them some encouragement and to survive this pandemic.
Yes.
And, so thank you so much again. We'll meet. We will meet again in another forum. And meanwhile,
Sure.
I'll pass to our secretary, our MC.
Sure.
Renu.
Thank you very much, Professor Kalimadi, for the informative talk. And thank you very much Senior Professor M. Ravichandran, for moderating the session. On behalf of ABSA and the Webinar Organizing Committee, I would like to take this opportunity to thank Professor Kalaimadi for the enlightening talk on SARS-CoV-2 vaccines. We are very grateful for the time and effort you have taken to share your thoughts and experiences with us. A master moderator helps shape a session, and our session today was indeed an insightful one. Thank you, Senior Professor M. Ravichandran, for the terrific and very professional example you set as a moderator. Thank you to all the participants for joining us in this webinar. Besides, I want to thank the organizing team for their efforts to make this webinar a success. Once again, thank you everyone for making this webinar a success. Before ending the session, we would like to hear feedback from all participants by scanning the code on the screen right now. We promise the evaluation survey would not take you more than 1 minute. Your feedback will be greatly appreciated. You can either scan the QR code or go to the link shown below. The link is posted in the chat box as well. After the completion of the survey, you will receive an instant participation certificate in a short while, and I will keep this slide running for 2 more minutes for you to scan the QR code. Once everyone has scanned the QR code, we will have a photo session. We're looking forward to have all of you join us again in the next session. Thank you again, everyone, and we wish you good health and safety.
Thank you, Renu, for hosting this, and thank you, Prof, for moderating this session, and thank you all the audience for your patience and your kind comments, and really appreciate. Presentations are only worth if the audience are there. So you made the presentation, and thank you for the opportunity to share little I know. And good luck with everything, and stay safe, stay masked, and get vaccinated.
Stay here for some more time, Prof. We'll take a picture.
Sure, I'll do that.
I hope everyone have the chance to scan the QR code. Therefore, before we end the meeting today, we will have a photo session. Everyone, please turn on your camera, and I would like to have our protocol, Ms. Vinodini, take over for the photo session.
You want to unshare your screen?
Yeah.
One, two, three.
How are you taking a photo session? Everybody is...
Okay, done.
Everybody on the camera, please. Okay, thank you. More faces. Nice to see faces in the pandemic.
Okay, done.
Thank you.
Thank you.
Thank you, Prof. We'll catch up.
Okay, then I shall leave. And thank you.
Thank you.
Good night.
Good night.
Good day.
Thank you very much.
Good
once
day.
you,
Have a
Prof.
nice day.
Thank you very
Bye.
much once again, and see you soon in our next webinar. Stay safe and have a wonderful day.
Thank you.
Thank you, Prof. Ravi, for moderating the session.
Yeah, thank you so much for your support, Dr. ABSA. We can plan for another gathering with Prof. Kalai.
Yes, sure. Meanwhile, I will get to work on the list lah.
Yeah.
All the informative websites.
Yeah. It should be useful for everyone.
Yes.
Thank you. Thank you for the support. I think thank you for spending your holidays here. Yeah, I think it's indeed useful. Okay. Bye-bye.
Yeah. Bye, Prof. Thank you. Thank you everyone for coming.
Thank you for
For your
Thank you,
time.
Prof.
Yeah, thank you for others who joined from other part of the world. Our prayers are there. Stay safe. We are here to support everyone. And hopefully we'll heal out of this pandemic. Stay safe. Bye-bye.
Bye. Okay, so I shall end the session now. Till next time we meet everyone. Bye.